Hello everyone! Today we're going to be talking about the lactate dehydrogenase or LDH reaction. So LDH catalyzes the interconversion between lactate and pyruvate, and the direction of this reaction depends upon which of the substrates are present and also what the pH is of the surrounding medium. So enzymes including LDH are catalysts, and what this means is that they take a substrate and a cofactor, and for the case of this lab, this is lactate, is the substrate, and NAD plus is the cofactor. It brings them into close proximity, and this lowers the activation energy barrier for the reaction, and that allows the reaction to proceed more quickly than it would if lactate and NAD plus were to simply collide in a buffered solution. So the active site encompasses these two molecules, and what happens is that LDH has a catalytic histidine, and this histidine is deprotonated. So it has a set of lone pair electrons that can go and pluck a proton off the alcohol group on the lactate. So then that oxygen has an extra set of lone pair electrons that collapse down and then are added to the nicotinamide ring of NAD+. And because NAD plus has an extra pair of electrons now, it can pluck a hydrogen from the media. We consider this to be a hydride transfer because you have a hydrogen that's being transferred along with a set of electrons. And the result is we have a pyruvate molecule. So the oxygen on the lactate has been oxidized to a carbonyl. We have NADH because the NAD plus has picked up a set of electrons as well as a hydrogen. And then along with this, a proton is also released into the media. And so one of the results of this is of the reaction producing pyruvate, NADH, and H+, is that as the reaction proceeds, the concentration of protons in the media is also going to increase, which means as the reaction goes, we're going to see the pH of the solution decrease, and that's something we're going to keep an eye on and talk about in more detail throughout the semester. So I mentioned earlier that the direction of the LDH reaction depends on the pH, and in our lab, we monitor the lactate to pyruvate reaction at a pH of 10. So one of the questions that this begs is, why do we have to perform this reaction at a pH of 10? Well, it has to do with the pKa of the histidine, of the catalytic histidine. So for an amino acid, so at the pKa of around 6.7, this means 50% of our histidines are protonated. So if we draw out the histidine ring, so if we draw out the histidine ring here, that's a really sorry pentagon there. So 50% of the histidines are going to be protonated, and then 50% of the histidines are going to be deprotonated. And the 50% will have those lone electron pairs that are available to pluck a proton off of lactate. So if we lay this out, so at a pH of 6.7, half the LDH molecules are going to have the protonated histidine, and then the other half of the LDH molecules in solution are going to have a deprotonated histidine. So the way that we can shift and we can force LDH to either have all of its histidines protonated or deprotonated is by shifting the pH away from the pKa. So if we have a pH that's less than the pKa, we're going to shift the population to being mostly protonated. Whereas if we crank up the pH and make it greater than the pKa, so say a pH of 10, we're going to be favoring the deprotonated form. So at a pH of 10, we're going to see the deprotonated form, which means those lone pair electrons are free to pluck the proton off, a proton off of the lactate molecule. So what would happen if we were to change the pH? So if we change the pH and we added lactate and NAD+, nothing would happen. And that's because if we were to go ahead and if we had the active site and the histidine had a proton, or the histidine was protonated, it would not be able to get in there and pluck the proton. So nothing would happen. However, if we had pyruvate and NADH, we would see the reverse reaction occur. And in this reverse reaction, so if we were to draw out LDH on this side and draw our protonated histidine, what happened is that the electrons are the electrons from the carbonyl would actually pluck the proton off of the histidine molecule. And that will be the reverse reaction. So that would be the reaction, the pyruvate to lactate conversion that we would see at a pH of 6. But again, being at a low pH and having that protonated histidine blocks the lactate to pyruvate conversion. So that's why we need to have a pH of 10. So I'm talking about LDH here, and you may be wondering, why do we care about LDH? So very briefly, the energy in our body, so the production of ATP, comes from glycolysis, which produces 2 ATP, some electron carriers, and then 2 pyruvate molecules. And in normal cellular respiration, the pyruvate through the citric acid cycle and oxidative phosphorylation is broken down into CO2, water, NAD+, and then 36 ATP energy molecules. We're not going to talk about anaerobic alcoholic fermentation because we're not concerned for that for this lab. So aerobic oxidation, which concludes with oxidative phosphorylation, can only happen in the presence of oxygen. So what happens when there isn't any oxygen? Well, that's where lactate dehydrogenase comes into play. Because what happens is when there isn't any oxygen around, LDH kicks in and it takes all the pyruvate and turns it into lactate. And it does this because this generates NAD+, that is then shuttled back into glycolysis. And so as a result, we're only producing 2 ATP versus 38 ATP. So it takes a bit of an energy hit, but it allows the body to continue making energy when there isn't enough oxygen. A familiar case of this is when we exercise and the oxygen can't get to our muscles quickly enough. And so our muscles undergo this process instead, and your muscles start to ache because lactic acid is acidic and, well, acid burns. So, but another reason why we're concerned with LDH is because it turns out that cancer cells have developed the ability to undergo anaerobic homolactic fermentation, but they do this when there's plenty of oxygen present. And so because cancer cells are undergoing this version of glycolysis, but normal cells don't, this makes LDH a potential anti-cancer target. So as a way to differentiate cancer cells from normal cells, and then use that as a way to kill cancer cells, but without killing normal healthy cells. And we're going to touch a lot about that in the last lab of the semester. So now let's talk more about practically how we're going to measure LDH activity over the course of this lab and throughout the course of this semester, or at least the first half of this semester. And so the bigger question then is, how do we measure enzyme activity? So I mentioned earlier that enzymes work by bringing a substrate or this enzyme works by taking a substrate and a cofactor, bringing it into close proximity and enabling the chemical reaction to happen. So the velocity or the speed of the reaction is dependent on the concentrations of the substrate and the cofactor. So if we have a solution of substrate and cofactor, and initially we add in the enzyme, the reaction is going to proceed fairly quickly because if we have a lot of substrate and cofactor present, it's not going to be very difficult for the enzyme to reach out and grab the substrate and cofactor and make that reaction happen very quickly, assuming that the rate of the reaction is much greater than the rate of substrate cofactor binding. However, eventually, and so you can see that as the reaction progresses, the enzyme is producing more and more micromoles of product. However, what happens is eventually the enzyme is going to start running out of substrate and cofactor, and the amount of products are going to decrease. And so as the enzyme is running out of substrates, it's going to take it longer to find the substrate and the cofactor, and the enzyme's going to slow down until it eventually runs out of substrate and cofactor completely, and it'll poop out, or will reach the limit of detection for the spectrophotometer. And so this happens because as the course of the reaction goes, we run out of substrates. The enzyme is going to catalyze the reaction at different rates depending on the availability of the substrate and the cofactor. It'll also change if the products interact with the enzyme. That might change the rate as well. And so it's really important that when we're measuring the activity of the enzyme, we want to hone in and get a snapshot of the enzyme when it has a large amount of substrates and cofactors, and ideally the reaction rate will stay constant through the whole time we monitor it. And so in order to achieve this, we need to have a concentration of enzyme that is much less than the substrates. And the goal is that by having the concentration of enzyme being lower than the substrates, and if we want to be able to monitor the enzyme reaction at a constant velocity, so it turns over the chemical reaction at the same speed throughout the entire time we're monitoring it, we would want to manipulate our system so that our chemical reaction is a straight line throughout the entire time we watch it. So we're not seeing the enzyme act differently at the beginning versus the end of our chemical reaction, which is what we're seeing in this particular circumstance. And so the way that we can do that is we can manipulate the concentration of enzyme relative to the substrates. Because it turns out that at least in the linear range, the concentration of enzyme is proportional to the rate of the reaction. So if this is our enzyme reaction at concentration X, the rate of the reaction at 1/2 X will be half the rate of the reaction, and if we double the concentration, the rate will be twice. The rate will be twice as high, or twice as large. And the way that we can measure the rate from the micromoles of product formed per minute is by taking the slope. So the change in the micromole of product formed and the change in time. So the micromoles of product formed per minute. And so again, thinking back to algebra and calculus, if we have, a line implies that the rate is the same throughout the entire reaction, whereas if we have a curve and the slope is not staying constant, our enzyme is catalyzing the reaction at different speeds at different times of our assay, which is what we don't want to see. So that's all fine and good, but practically speaking, how are we going to measure the LDH reaction? Because what you're going to get during this lab is you're going to get a solution of LDH that has been taken from a cow heart. It's not just LDH, it's LDH and a bunch of proteins. And it's not like in the chemistry lab where you weigh out the amount of LDH and you know how much exactly in there. You actually have no idea how much LDH is present in the sample. And so we can use the rate at which LDH catalyzes the reaction to actually quantify how much LDH is present. So first of all, how do we measure the rate of the LDH reaction? And that's with the velocity. Or we measure, so practically we're measuring NADH, pyruvate, and H plus production. But we do this by measuring the slope of the products formed against time. And so we need to measure LDH activity. So yes, we can measure the reaction velocity and see if it's where it should be, but we can also use the LDH activity to find LDH. And that's going to be really important during our purification process. So if one sample has activity and another doesn't, we know which one contains LDH. But we can also use it to quantify the amount of LDH in a sample. And so the way that we can quantify the amount of LDH is by determining the units of LDH in our sample. The definition of a unit of LDH is it's the amount of LDH that can produce 1 micromole of NADH and pyruvate per minute. Or 1 micromole of products per minute. So we can use the rate at which the enzyme catalyzes the reaction, so the micromoles of products that are produced per minute, to figure out how much LDH is located in our sample. And the way that we actually measure this is pretty tricky because we can't just look at the spectrophotometer and see pyruvate NADH and protons being produced. We have to visualize them in a tricksy kind of way. Because if we take a look at our products, so if we were to take NADH NAD plus and NADH and H plus, and we were to throw them in a UV visible spectrophotometer and scan them for absorbance as a function of wavelength, what we would find is that at a wavelength of 340 nanometers, NADH in the dashed line has an absorbance, but NAD plus does not. It does not. So what this means is that if we were to watch our cuvette as a function of time, the absorbance at 340 nanometers is going to increase as more NADH is produced. And so that's exactly what we do. So if we look at the absorbance at 340 nanometers as a function of time, as NADH is produced, the absorbance at 340 nanometers is also going to increase. And if we've done our enzyme dilutions correctly, this will be linear. And again, the slope of the change in absorbance at 340 nanometers gives us the velocity of the reaction. And I'll show you in a little bit how you can convert the absorbance into micromoles of product. But first, let's take a quick review of spectroscopy. So what happens in our UV visible spectrophotometer is that we have a lamp that gives off light over a variety of wavelengths, and then we use the monochromator, probably mispronounced that, to select a wavelength at 340 nanometers or any other wavelength choice. And so that way, only light at a wavelength of 340 nanometers is sent through the cuvette. And so what happens is if we have NADH in our cuvette, the light is going to be absorbed and less light is going to be transmitted to the detector. And as a result, we're going to see a big absorbance. So if less light is transmitted, that results in a bigger absorbance reading on the detector. Whereas if NAD plus is present, like at the beginning of our reaction, that light is going to pass through, it's going to be transmitted through the cuvette onto the detector. And as a result, we're going to see a small or no absorbance at all. And so so this would be an instance of 0% transmittance, and this would be an instance of 100% transmittance. And then we can use the Beer-Lambert law, more on that in a second, to convert the change in absorbance, to interconvert between the change in absorbance and the change in the concentration. So the instrument that we're going to be using is a Carry 60. This is a nice instrument because it records all your data for you and in very short increments, and it has a computer hookup. And the data that you generate are going to look like this. And so the portion down here comes from your blank where you have your cocktail. So that's just your lactate and your NAD plus or your NAD plus. Then at this point, you're going to add LDH. And after LDH is added, you'll notice that there's this beautiful line over the course of 2 minutes. And the slope of this line, which is the change in absorbance per minute, that's going to give us that can be converted into the velocity of the reaction. And then once we get our slope, so our change in absorbance per minute, we can use the Beer-Lambert law to convert the change in absorbance to change in absorbance per minute to the micromoles of products produced per minute. And so if we have the Beer-Lambert law, where A is our absorbance, epsilon is the extinction coefficient, and remember this is a constant for a particular compound at a particular wavelength. And so for NADH, which is what we're detecting, this is an extinction coefficient of 6,220 molar to the -1 centimeter to the -1. C is the concentration. Rather, remember this is a rate, actually. That's micromole, or that's micromole. It's going to be a rate, micromole per minute. And then L, or B, also written as B, is the path length. It will depend on the cuvette used, but for our lab, this is going to be 1 centimeter. Don't get too comfortable with using 1 centimeter because I'm perfectly capable of switching that up in the practice problems. And if we rearrange, we get C equals A over epsilon L. And so you can C equals A over epsilon L. And so we get the delta A per minute divided by molar to the -1, centimeter to the -1, times centimeters. Centimeters cancel, and you end up with molarity per minute. All right, so we've figured out how to quantify the rate in terms of molarity per minute. So how do we get to the number of enzyme activity units, which is quantified in terms of micromoles per minute? So we start with solving the Beer-Lambert law for the concentration. So we take the change in absorbance per minute. So change in absorbance, and that's a change in time, but we can say the change in absorbance per minute. We divide it by the extinction coefficient, so 6,220 molar to the -1 centimeter to the -1. And that gives us molar per minute. But our activity units are micromole per minute. So the next step is to convert this to micromole per minute. Then the next question is, so we have our rate in units of molarity, but enzyme activity is in units of moles or micromoles, not micromolarity. So the last step is to convert to micromoles, and we do that by multiplying by the volume of the reaction. So this is the volume of the reaction, or this is the volume in the cuvette that we actually measure. And so that's 1 ml of solution plus 25 microliters of enzyme that you spike into the cocktail. And that will give us a rate of micromole products produced per minute, or the units of enzyme in that cuvette. So now that we know how to figure out the enzyme activity units, how do we set up our enzyme assay so we find the correct amount the correct amount of LDH, and we can accurately quantify the units? So when you start your enzyme assay at the beginning, if you were to take the sample that we gave you and put 25 microliters right into the cuvette, you're likely going to see something like this. So you have your background, and then once you spike in your enzyme, the absorbance, and by extension, the concentration of the products is going to spike up and then flatten out as either the enzyme runs out of substrate or you saturate the detector. And so our delta A per minute is like 1. So that's pretty big. And what happens is this slope is too big to accurately quantify the rate of the reaction, and you're actually going to end up underestimating the rate, because again, we want to see the reaction be linear for 1 to 2 minutes, and we're shooting for a change in absorbance of like 0.2 to 0.3 A per minute. And so this would be a final absorbance of 0.6. Not clearly, so we want our reaction to look something like that, but clearly we're not, and we're going to end up with a value that's lower than it actually is. If you take your enzyme and there's too little enzyme that you add into the reaction, you might see a situation like this, where the curve is actually too shallow. It's too shallow to accurately, to get an accurate reading of the activity and then to figure out the units of enzyme that are in your solution. And so we can change the slope by increasing or decreasing the amount of enzyme. And we can do this by making appropriate dilutions of our stock, of our enzyme stock, before adding it to the cuvette. And that's exactly what you're going to end up doing. Now, to ensure that you make proper dilutions, you'll need to use the M1V1 equation. So now we're going to go through a few different practice problems. The first of which is how to make an enzyme dilution. All right, so let's say you're starting off in lab, and your TA suggests, hey, maybe you might want to start with the 20x or 1 to 20 or a 20-fold dilution, and you want to make sure that at the end of the reaction you have 150 microliters. How are you going to make that dilution? Why don't you take a minute, pause the video, and give this a try for yourself. All right, so with the dilution equation, we have M1 or M initial V1 equals M2 V2. So we know that we want our final volume to be 150 microliters. And the concentration of our stock is 20x, or it's 20 times more concentrated than our 1x or our final solution. So we have to solve for how much of the 20x we need to add to make a 1x solution of 150 microliters. And so we take V1 is equal to 150 microliters divided by 20, and that gives us 7.5 microliters. So we'll add 7.5 microliters of 20x and 150 minus 7.5. And so that means we'd add 142.5 microliters of buffer. All right. So let's try another problem now. So let's take a stab at measuring how many enzyme units are located in a particular enzyme reaction that you've run. So from fitting data on a previous page, you determined that the change in the absorbance was 0.264 absorbance units per minute. So that's right in the sweet spot, that nice linear range. We're going to change this for easy math, a 1 ml assay when you use 25 microliters of a sample that have been diluted 1 to 20. How many units of LDH are present? Note that the extinction coefficient for NADH is 340 nanometers is 6220 molar to the -1 centimeter to the -1, and the path length is equal to 1 centimeter. Why don't you go ahead and pause and try this calculation out for yourself. All right, so the first thing we have to do is we have to convert absorbance to concentration. And we do that by taking the change in absorbance per minute, so 2.264 A per minute, and we divide by the extinction coefficient and times the path length. So 6,200 molar to the -1 centimeter to the -1 times 1 centimeter. Let's cancel. And so we know that this gives us molar per minute. So next we have to convert molar per minute to micromolar per minute. So we do that by multiplying by 10 to the 6th micromole per mole. Then, after we've converted from micromolar to molar, we then have to convert micromolar to micromoles. And we do that by multiplying by the volume of the total reaction in the cuvette. And so the total volume in the cuvette is going to be equal to 1 ml plus the 25 microliters of enzyme we added, or 1.025 mLs, which is equal to 0.001025 liters. We multiply by liters, 0.001025 liters, and that's going to give us 0.044 micromoles per minute, or 0.044 units. All right, so the micromole, so when we calculate the units of enzyme, that's telling us the amount of enzyme that's located in the reaction cuvette. However, the amount of enzyme that's in the cuvette doesn't tell us anything about how much enzyme is actually located in our big sample that we pulled from, because we haven't taken the dilution factor into account. And the way that we do that is by calculating the relative activity. And so that's really important, especially if you want to compare your sample to somebody else's in the lab, because you may end up with totally different units of enzyme or a different activity because you guys use different dilutions, or maybe you started off with initial samples that had different concentrations. So calculating the relative activity allows you to figure out how much LDH is in your starting sample, but it also allows you to compare your results to another group's results. And the way that we calculate the relative activity is by taking the units in the assay, and then you divide it by the milliliters of enzyme that you added to the assay. So this isn't the volume in the cuvette. It's the volume that you added to the cuvette that contains enzyme. So that would be the 25 microliters, or the 0.025 mils, and then you multiply it by the dilution factor. So now I want you to take a minute and I want you to calculate what the relative activity is of the initial sample, of the original sample. And that's if we have 25 microliters of a 20-fold dilution sample. So we know from the previous problem that we have 0.044 units, and we added 25 microliters of sample, so 25 microliters, do the conversion here, it's 1,000 microliters to a milliliter, and that gives us 0.025 milliliters. So we divide by 0.025 milliliters, and then we multiply by the dilution factor, which is 20. It's 20, and that gives us take a minute and calculate this. So that gives us a relative activity of 35.2 units per ml. I know this is a lot of math and there's a lot of units being thrown around here, but one way to check to see if you're doing the right thing is to see if your units match up with the units of the quantity that you're trying to calculate. So are you getting units per mil for your activity? When you're calculating your units, are you getting an answer on micromole per minute? And in order to do that, you need to make sure that you're keeping track of your units as you do your calculations and writing them down. And so this is something that I want you guys to be thinking about. So in this lab, you're going to be determining what the effect of the concentration of CAPS buffer is on the reaction rate of LDH. So I want you to take a minute and I want you to think about which of these buffers, 150, 15, or 1.5 millimolar CAPS, is going to be the most effective and why. I want you to take note of this and I want you to see if your hypothesis was correct based off of what you see in lab. All right, and so this is miscellaneous information we'll cover when we actually get into class. So good luck with your pre-lab assignment, and please feel free to shoot me an email if you have any questions.